So I'll be passing the mic very shortly over to Dave Rosen, who's going to chair most of the day. 23 years ago when we started this, we had, I think maybe 30 people came. We had a small room, we had slides and overheads. And today, of course, we're using technology of all sorts, including our registration today has gone over, I think, 220 people. This room doesn't have enough seats, so we're scrambling here, and we've got a webcam that's streaming to another room. And of course, nothing's compatible. And every one of you that may be streaming stuff off your phones is taking away our bandwidth as well. And so we're all trying to make this work today. It's an experiment. We didn't want to prevent people who wanted to come from coming. And so we've tried this as a solution. Let's see, any special instructions? Presentations are designed to be short, geared to be 5 minutes. And as Mark, as my production manager, is pointing out, you got to stand in front of the camera. So our speakers will please stand back here. You do have a monitor here where you can see exactly what's being shown. And because my voice isn't very good, when you exceed 5 minutes, you'll see the chicken. And if you don't see it, you're going to hear it. A few people have experienced the chicken. They've been scarred for life. But they're still coming back to speak, which is good. I'm now going to pass the mic over to Dave. I don't think we have any special announcements other than washrooms are out in the entryway. Please don't bring food into the room. If you had come intending to speak today and are not on our list, let us know. We'll get you loaded. We do have time on the agenda. And we've got lots of time at breaks and at lunch for people to talk. Finally, you can, if you want, at the lunch break, go over and see Big Blue. The museum will allow everybody who's here today to get in for free to go and have a look at her. And pass it over to Dave. And you may, there's also a mic here on the podium.
I'm just going to use the podium mic. So hopefully everyone can hear that because this is what the speakers will be using. The only thing I do want to point out, we have an overflow and we got people crying on the stairs. We actually have about a dozen seats still scattered around if people want to, particularly over by the wall. You know, typical architecture failure where the seats are over by the wall, so you either have to push through or climb over the wall, like invading forces, but either one is fine with me. So we have quite a packed agenda, so we're going to start off right away with Brianna Wright is going to be speaking about southern resident killer whales. Northern. Oh, right, there are no southern. I forgot.
What do I do to advance? This guy?
Oh, yeah, sorry. Thanks. So we do have, you can use the space bar on the computer, or we do have a remote. Do not use the remote to travel, or they're going to get very mad at you and give you stage directions back to stand here. I will be yelled at, I'm sure, when it's mine.
Okay, hi everyone. I guess I'll start this off by saying the reason the title of this talk is so long is it's a little bit of shameless self-promotion on our part because we've got a paper under review on this topic. So hopefully in a few months, you'll be able to read more about it instead of just 5 minutes. This will be the sort of Cliff Notes version. So I think everyone's pretty familiar with all the photo ID work that's been going on this coast for resident killer whales over the past 40 years or so. And one of the most interesting things that came out of this work in terms of how social structure is organized in killer whales is that neither sex ever leaves the matriline that they're born into. So they're one of the most highly philopatric, or stay with the family group animal that exists. Basically, pilot whales and a couple species of bats are the only other animals where this has sort of been described. So it's highly, highly unusual. Part of the reason for this is that usually one sex or the other will disperse because you've got an inbreeding problem. If you stay close to your family and males and females come into sexual maturity and they're related, that's a problem. So killer whales avoid this by mating non-locally. And they are able to do this through kin recognition related to their acoustics. So individual acoustic recognition and also group acoustic recognition. So that's how they tell who's who and who's not a relative and who is. The other problem with never leaving your family is that it's going to bring you into competition with your relatives, and it's always preferable to be in competition with non-relatives who don't share your DNA instead of damaging, potentially damaging a relative that lives close by. So that sort of begs the question, if this creates all these issues, why would it evolve? Obviously, it has to have a benefit that outweighs all these potential problems. And the general answer to that is that you're probably in some way helping your relatives. So in addition to reproducing yourself, you're helping your relatives to reproduce and survive, and that increases your overall fitness because your DNA is shared with everyone and is being passed on. So looking at this, one of the common helping behaviors that we've seen with resident killer whales that became clear over the years was one of food sharing. So these guys will, might need help with this one, Dave, sorry. I just need to click on the screen and I can't work that trackpad. Yeah, technical difficulties. Anyway, so we've collected about 12 years of data, 2002 to 2014, of prey sharing behavior where we watched these animals take salmon, break them apart, and then share them with one another.
Okay. I don't know what happened to your mouse.
Yeah. I pressed this button before.
No, it's because it's Here we go.
There we go.
I still have no mouse.
It is, yeah.
I was trying to do.
We even tested this.
I don't get it.
Okay, we can skip it. Sure. Yeah. Okay, we'll move on. Okay, instead of a really cool video, I'm going to show you really cool still shots. And these are from Barrett-Lennard's study out of the Vancouver Aquarium, and they were kind enough to share this with us. And Noah was also part of that study. And you can see, these are actually southern residents, but you can see they're L91, I think it is, at the front, and she's got a fish in her mouth and the little calf beside her. And then a frame later, you see her kind of turn back towards these other guys. And she's still got the fish, but there's also, it looks like a piece in the water that's broken off, and this other whale's making a beeline for it. So that's quite a common sort of behavioral cues that we see when they're sharing, is that they'll join up like this and end up breaking this fish apart and sharing it. So given that we're looking at a helping behavior, we expect that it'll be directed probably towards kin because of this close-knit family group, and that it'll be directed more towards very close kin. So we used a generalized linear mixed effect models for the geeks out there. That was our approach, but we'll move on quickly because probably most people don't care. So a few of the questions we wanted to answer is prey sharing kin directed? Is it within the matriline more so than without the matriline? Are close relatives favored over distant ones? So if you're looking to help relatives pass on DNA, it makes sense to help close relatives like siblings and parents than it does to help a cousin. We also wanted to look at whether some age sex classes shared more than others, and Graham and John did a paper in 2006 that sort of supplied some information that suggested that. So we just wanted to reconfirm that, and then whether certain age sex classes shared with females, with males, subadults. And then another question that arose is because a lot of the prey sharing we were seeing was mothers sharing with their offspring, we wanted to know if it was just across all offspring equally or if there was something driving that. Was it sex of offspring, age, birth order? Did that affect prey sharing at all? So here's some of the results. So I'll just try and go through this quickly so I don't get chickened. So we found that adult females are sharing pretty much everything they're catching, and a lot of it is directed towards their offspring, which makes sense to have those offspring survive. But it's a widespread behavior. It occurs with males, it occurs with subadults, so it's spread throughout the population. We found that it was primarily within the matriline, almost 90%, so that sort of backed up our theory that this is a kin selection behavior. We also found that adult males shared prey a lot less often, and this kind of makes sense if you think about their social structure, because adult males, their offspring are not within their matriline, they're somewhere else with their mothers. So on average, an adult male is less related to his matriline than an adult female. We did find an interesting pattern with adult males sharing, though, in that they share a lot with post-reproductive females, who are most often their mothers. And this is quite fascinating because it's not kin selection. If you share with an animal that can't reproduce, it's not a kin selective behavior. So why would you do it? And the answer sort of comes through something called pseudo reciprocity. And basically, it's a behavior that appears to be altruistic, but is in fact underlying it, it's sort of selfish. So it's basically they're sharing with their moms because moms are sharing with them and increasing their ability to reproduce and survive. So it's sort of like you're helping mom out so that you get your benefit back. And it makes sense for a post-reproductive mother to invest in a son over a daughter, because over the lifetime, if the son is a successful reproducer, he's going to produce more grand offspring than a daughter will. And also, it doesn't increase the amount of competition inside the matriline. So a male producing offspring in other matrilines doesn't increase the amount of food that then needs to be supplied for the matriline of that grandmother. So because we saw this sort of favoritism towards sharing with sons from post-reproductive mothers, we looked at how sharing with offspring changes with offspring age. And we ran a model on this, and one of the most interesting things that came out was that daughters, gradually that sharing drops off as they age. So you can see on this slide, you can see that as daughters hit around 10 or so, the number of times where sharing is yes just disappears, and they're not shared with by mothers. And this is the model. So it sort of starts out really high when daughters are young and they're subadults, and then it drops off as they mature, and the moms just kind of stop sharing with them. And the time that happens is sort of about the time that these daughters are becoming reproductively mature. So one of the predictive factors that was really significant was when that daughter produced her first calf, her mother just kind of stopped sharing with her. And it's probably not a neglectful thing on the part of the mother. It's probably more related to the fact that the daughter now has offspring to care for. She's sharing with them and less so with the mom. And so it's sort of more related to that. So part of the really interesting story that came out of that is that if you lose that benefit of prey sharing, then what's your incentive to stay with your group? What's your incentive to be philopatric? It sort of starts to disappear. And that's not to say that all daughters are going to take their subgroup and leave as soon as they produce their own offspring, but we have been seeing a pattern where eventually, the oldest daughter's subgroup will start to fission off from the main group. And this is Eva's thesis. So she's here today. If you have any interesting things you want to ask about matriline fission, she's the person to ask. But this might be a contributing factor to that pattern that we're seeing. I have a lot of people to thank, especially all the people up top there that gathered information about prey sharing and collected observations in the field for us. That was a huge part of this project. Thanks.
Brianna's fleeing before a question. Yeah, we have time for a question before Rob gets the.
It's based on over 600, and those are filtered. So we filtered out ones where we weren't certain about who got what, or who got the fish, or who was sharing it. So it had to be a prey sharing event where we had one whale sharing with one or more other whales, and we had to definitively know the identity of all of them. Yeah.
Our next speaker needs no introduction, but I will. Anyways, John. Go one bigger and better, so he's going to be promoting his book and talking about some blue whale science.
Good morning. As Dave mentioned, last year up here I took 5, maybe 10 minutes of shameless self-promotion of my new book at the time. It's now a year old. I'm not even going to mention the book this year. Or the fact that you can get it over the bookstore. Instead, what I'm going to do is take one of the species of cetaceans in the book and just give you a little 4 minutes now on that species. I figured 25 species of cetaceans in British Columbia. I'm good if I do one a year. I'm good for a quarter century of this. So, all right. The reason I thought we'd talk about blue whales is because we've actually accumulated a little bit of information finally on these animals. This was a good year this year. And who's we? Well, this is the Cetacean Research Program team at the Pacific Biological Station. Wonderful team. They have all contributed to this work, as have many of you in the audience here who've managed to come out and work with us on surveys and so on. So not to forget, of course, the indomitable man on the West Coast, Brian Gisborne, who's sitting at the back there, just came in off the water last night, raced here to be here. So you got a lot of credit for Brian because he's out there looking for these things all the time. Okay, blue whales. I don't think I need to introduce blue whales. They're really big. They were hunted on our coast ruthlessly, of course, in the early days of whaling, started around 1907 or so. And you can just see how huge the animals are. There's people sitting on it there up at Rose Harbor. They were the most desirable whales in the whaling era. They're in the blue line there, starting in 1907. I left humpbacks in this map just to show you the relative number of animals taken per year. They were hammered really early on, and then they just kind of sputtered out. And were because they were so valuable, the whaling was sustained with other species like sei whales and fin whales. But whenever a blue whale was seen, it was hunted. And so by the end of the whaling era, here's one in 1950. They were getting really scarce by then. The late 1950s, color film was invented by then. There's a blue whale in color at the Cole Harbor Whaling Station. They were pretty much gone. They were finally protected in 1965, and they were one of the most rare species remaining. Okay, catches, that's about 270 where positions were recorded. This is where they were hunted along our coast. And this is the cumulative number of sightings that we have had, confirmed sightings since whaling ended. So not a lot, but they are out there. And we're starting to use, of course, photo identification to track individuals. This is the first one was photographed by Randy Burke and crew on the island Romer off South Moresby back in 1997, in June 12th to be exact. Interestingly, through our collaborators, John Calambakidis and Cascadia Research, they maintain the big catalog of California blue whales. It matched to that catalog, and 28 days later, it was down in Santa Barbara Channel off Southern California. So these animals are really mobile. They can cover huge distances at once, which is one of the reasons they're so hard to find. You never know where they're going to be because tomorrow they can be a long ways away. Here's another match between two animals, one seen in 2003 off Haida Gwaii, and then again down California. We don't have time to examine the patches, pigment patches on there to show that it's really a match, but it is. And since then, we've had a few more matches. So this is, as I mentioned, a big year for blue whales. That's a nice photograph of a blue whale from Speed Air, the little DFO surveillance plane back in early August this year. You can see that distinctive coloration, but also the shape of the rostrum is kind of rounded on the bow there, unlike fin whales, for example. So very distinctive shape there. As of September, with the latest photo ID, there's now 26 individuals, unique individuals in our catalog. This is actually a calf in the foreground traveling with its mother. If you look closely, you can see some killer whale teeth rake marks up on the dorsal fin. Here's a discovery curve, the cumulative number of unique individuals on the vertical axis there, and then years on the bottom. You can see it's pretty steep still because we're not getting any resites. There's plateaus when we've gone out on the water and not seen any blue whales the whole season. But of 25 animals that have been examined by John Kalmbakitis and crew down at Cascadia, 8 of them have matched to California. One individual down, you can see the little red dot down as far as Baja California. And so that matching rate relative to within the population of California is actually pretty low. So it looks like some of the animals up here are part of that population and some don't seem to use California. So we don't know where these animals go. So there's lots to be learned about blue whales. We also are listening for them with our network of autonomous recorders that are deployed offshore or with a shore-based facility up at Langara Island. If you look at a spectrogram of just one of these recordings, this one's from Bowie in January a couple of years ago. And I don't have a pointer, I can't show it, but the horizontal lines, they come in 2 pairs there, you can see. Those are blue whale calls. The A call is the longer one on the left, and the B is the shorter one. And they're kind of ubiquitous in these recordings. They're very loud, so we're not sure how far away the animals are, but it's not, they're present a lot of the time on the recordings through from summertime right through into at least January. All right, last year, February of 2014, our friends Stan and Karen on the North Coast discovered a dead whale rotting quietly in a bay off the west side of Banks Island, which is on the north coast of BC. And they sent some photographs down, and we looked at this and said, oh, it must be another dead fin whale. Pretty big one, but a dead fin whale nonetheless. Then we looked more carefully at the pictures, and we said, my God, look at the head, the shape of that rostrum. It's got to be a blue whale. What's a blue whale doing dead in a little bay on Banks Island? And there it is later. You can see distinctive black baleen, characteristic of blue whales. And it was indeed a big blue whale. And by the time they got back in March, the carcass had mostly gone except for the mandible, one of the mandibles, and the skull here. So the museum in Victoria was all excited about getting this skull. And through really epic, heroic work, here Stan lassoing it, and efforts by James here, wading through the festering water with this mandible. They got it all trussed up with floats and managed to float this giant thing. How long was it, James? 20 feet or something? 18 feet. And got it all set, and it was getting towed down towards civilization when a freak storm came up, flipped it out of the boat, and it floated off. So it's sitting on a beach somewhere, if anybody finds it. All right, that's what I want to talk about. Thank you. Question.
Over here.
Sorry.
Stand here.
There's no
Questions
questions.
for John?
No.
Oh, it's perfect. Okay. Thank you. Our next, Adrienne's going to be our next speaker. He's going to be telling us about baleen and showing us. I hope that's what you've got in the bag.
Okay, so yes, so I study baleen, so I have some here, and so it's actually upside down there. It grows down from the top jaw, but basically 2 kind of important parts of baleen to differentiate between are there's tubules and there's bristles. So the bristles are what are exposed here from the matrix, and so as this matrix wears away, these bristles become exposed, but they're actually continuous, so they start all the way down at the base of the baleen where it grows from. And then when they're enclosed in this matrix, they're known as tubules. And then as they get exposed, they're called bristles. And so these bristles, not a whole lot is known about them and how they relate to the whole filtration process in general. But one observation that I'm kind of basing my study off of is that they taper. And we don't know why, but I'm kind of using this observation as an opportunity to learn about their growth. So basically, there's 2 hypotheses that I'm differentiating between. So the first is that if tubules taper, then the taper of these bristles is a result of their growth, and that's just something that they have throughout their whole life. Or alternatively, if the tubules don't taper, and that's something that only the bristles possess, then that means that it's probably degradation that's causing these bristles to taper. And so that might be changing their properties in some way. So first, I've confirmed that bristles do in fact taper. So this graph is just showing the bristles at 3 different points from their base to their tip, and just showing that the bristle perimeter is indeed significantly decreasing. So, okay, so the bristles do taper, and then by taking cross sections, I've looked at the tubule taper in the plate, and I've determined that there's no significant taper. So their perimeter is pretty much consistent all the way through the baleen plate until it's exposed. And so the way I did that was just by taking 3 cross sections, so from the base all the way down to right before the tubules are exposed as bristles. So you can see that picture there is from the left to right, the tubules all the way through. And so I can actually track the same tubule all the way through and then see how its perimeter is changing. And so that's what I've done. So by determining they don't taper, I realized that my second hypothesis is supported. So it's degradation that's causing this bristle taper. So knowing that, I kind of have a few next questions that I'm addressing that I'm currently investigating. So I know that these bristles are degrading, but I don't know what effect this has on their mechanical properties. Basically, I'm wondering if bristles that are more degraded, are they actually weaker? And so I kind of have some predictions of different degradation within baleen whales. So there's 2 main types of feeding styles. So the one on the left there, these whales they basically take a massive gulp of water and then they kind of forcefully expel it through the baleen. And that takes a lot more energy and a lot more force than in contrast to the other style. So it's more relaxed, I guess, and they just kind of open their mouth and let the water flow through the baleen in a more unidirectional fashion. So there's some pretty big differences between those feeding styles. And as you can see, so the baleen on the right, or on the left, that's the one I have here, and so that's from a fin whale, and that's the more energy-intensive style. So you can see that the bristles are a lot thicker and they're a lot shorter than those in the one on the right. So those are from the more relaxed style. So that's from a bowhead whale, which is one of the organisms I'm studying. And so basically I can see that the baleen itself, it's very different in morphology, and I'm guessing that there's different patterns of degradation within these species. And so I'm going to see if whether the mechanical properties of the bristles themselves, if there's actually any differences. And so knowing that, I can kind of maybe help to determine the function of the bristles in general and see if they actually have any advantage to the feeding process or if they're just kind of just an unavoidable consequence to the wear and the degradation of the plate itself. So that's where I'm kind of at right now. So I've determined that tubules, they don't taper, bristles are probably degrading, and then now I'm currently investigating what effect this degradation has on the bristles' tensile properties. Yeah.
Any questions for Adrienne?
Pardon me, what age the bristles start being produced? I think they get produced pretty quickly, and the baleen itself, like as the mouth grows, more baleen plates appear, but the bristles are pretty much always exposed. Yeah.
So I presume there's a small typo in the agenda, or are you going to tell us about
Yeah, I didn't study a whole year.
a whole year? That's an amazing talk.
That would be a lot longer than
Okay.
5 minutes.
All right.
So I'm just going to jump in with a little background here. You'll see from these numbers that the Beluga Hearing covers a much wider range of frequencies than humans, as well as a much higher range. And basically, sound waves enter through the jaw and that region and are transferred to the middle ear and then on into the inner ear. And here's where we find the cochlea. Now that's a small spiral-shaped structure which is fluid-filled and contains 3 ducts or channels, the central one of which contains what's known as the organ of Corti. If you'll notice the picture on the right, there are some purple structures there, and those are hair cells, of which there are 2 types, the inner hair cells, or IHC, and they form a single row. And then we also have the outer hair cells, or OHC, which are arranged in 3 rows. Overlying that is a layer of cells called the reticular lamina, and protruding through those are stereocilia, which are attached to the hair cells, and those are the structures we're going to be looking at most today. So you'll see here a flattened version of the cochlea if we uncoiled it and laid it out, and there are a number of morphometric and morphological changes that occur as we progress from the base all the way to the apex. And along with those morphological changes are changes in frequencies that are detected at those locations. So you can see things such as spacing of cells, the height of the stereocilia, and so on, all change. So why is this important? We know that the frequencies received in the cochlea at the base, higher frequencies are detected, and then as we move towards the apex, low frequencies are detected there. So if we can correlate those frequencies with their location, we can develop a frequency map for belugas. So for instance, if we found acoustic trauma in this region, we could potentially associate it with the sound that caused that. We can do that throughout the cochlea. So the basis for the methodology we were looking at is from a paper by Yaron, looking at guinea pig cochlea. And you might think, guinea pigs, they're tiny. Belugas are much, much larger. Where is the similarity? But there are many well-documented trends in mammals relating the morphological differences to the frequencies at any given point in the cochlea and how those change throughout the cochlea from the base to the apex. So the objective of the work I was looking at was to determine which of these morphometric features are frequency dependent within the inner ear of the beluga whale. Our samples were obtained through legal subsistence hunting, which allows for a fresher sample, less degradation, and then in turn, more reliable results in the end. So we started out using a scanning electron microscope to take micrographs of the cochlea and used ImageJ software to analyze those photographs to determine the measure of the cochlear length as well as morphological parameters. So we started taking the micrographs, and as we take each one, you have to tilt the sample to accommodate for the curvature of the cochlea. I looked at 120 photographs similar to this to measure out the area. Each one has to correspond to the next photo to ensure an accurate length. And our cochlea was about 38 millimeters in the end. So the next step was looking at 6 different locations around the cochlea to further analyze some of those parameters that I discussed. These are the 7 parameters that we analyzed. We did multiple measurements of each at each of those 6 locations, and we found trends in 4 of these that match the trends that we have well documented in terrestrial mammals. So you're going to see a few graphs, and just the horizontal axis is the distance from the apex. So we move from the apex to the base, and the y-axis here is, in this case, the parameter of the inner hair cell, that single row, all the way to that 3rd row of outer hair cells. So you can see here in the beluga, there's a decreasing trend. So as we move from the apex to the base, that distance decreases. And if we overlay the work from Yaron in the guinea pig, we see that there is a correlation among the frequencies. Now, in this graph, as well as subsequent ones, you're going to see that I have data points beyond the range of the guinea pig. So I'm going to touch on that later, but the guinea pig upper hearing limit is only 30 kilohertz, so that makes sense, but the trends are in agreement. So these are the other trends we saw. So again, the first graph we just looked at, and we also see a decrease between the gaps in that 2nd row of cells, outer hair cells, and the 3rd row of outer hair cells. So they all decrease as we move to the base. And then in the width of the outer hair cell, that actually increases. And these are all trends that are consistent in land mammals. And again, looking at the guinea pig, we've overlaid the frequencies, and again, as I touched on, there's the gaps, but I'm going to talk about that in a minute. So we feel that the current results are promising and preliminary, but show some potential. We want to do more research on numerous individuals, additional locations within the cochlea, as well as additional morphometrics. So just to move forward, where are we going to go from here as far as figuring out those gaps that I talked about? This is just showing you the frequency range for humans and then guinea pigs, which is a little more than ours. And then you can see the area of overlap between the guinea pig and the beluga. And then the full range of beluga hearing extends quite a bit further. So how are we going to figure out where those gaps are? We're going to look at echolocating bats. They have a similar hearing system as far as echolocation as in belugas, and they cover a similar hearing range. So potentially we can use that to fill in some of the gaps and make a full map. And this could also be a suitable alternative for traditional frequency mapping techniques. And I would like to acknowledge and thank these people without whom this research would not have been possible. Thank you.
Questions for Cassandra? You're okay. Thank you, Cassandra. Next speaker is going to be Paul Cottrell, who's going to tell us about the about this response network. I was going to call it Stranding Network. It's response network.
Good morning, everyone. I have, in fact, been scarred by the chicken a couple years ago, so I'm definitely going to keep my talk within the 5 minutes. And also, Pamela said she's going to beat me with that chicken if I go over. So what I want to do today is just kind of talk on some of the highlights, but also really just thank the many, many, many organizations, individuals, researchers, academia, NGOs that really make this program work. Clicker here. I also have video, so I'm hoping that's going to work because it's a big part of my presentation. Yeah. Start. I think everybody's probably aware of the BC Marine Mammal Response Network. Again, we've had a stellar year in terms of the number of incidents reported. And I think the 1-800 number, and I brought lots of stickers and information on that, it's so important for this program for individuals that do see injured, distressed, entangled marine mammals to phone right away. And I think every year we've been getting better at that and more calls that are timely. So we've been, it's really helping our success rate. Because of financial restrictions and resource limitations, we focus on species at risk, obviously the threatened and endangered, and also species of concern. And our success rate for response, whether it's pictures or species ID or actual on-scene response, is quite high for species at risk. Our volunteer network and groups that help is coast-wide these days. And so we're really happy about that. And of course, we have individuals that, Dr. Ravery, for example, we've really called upon this year because of the large number of large whale deaths. So it's, and I'll be going through some of that as well as some of the number of whale entanglements that we've had this year. So we're well over 500 incidents already this year, and we're kind of in line with the last couple years. And this program was initiated in 2008, and Lisa Spaven is just an amazing start to this program and really developed this program into what it is. So without Lisa, this program would not be where it's at. So I want to acknowledge Lisa. Quickly, just going through the strandings, the large whale strandings, we've had a huge number of fin whales this year. We've had 5, 3 in the last couple weeks up in Bella Bella, all together on the same beach. So along with that, we've had 6 humpback whales. For the fin whales, we were able to do necropsies on all 5. For the humpback whales, we were able to get out and sample 3 of those. We've also had a dead sperm whale and a dead gray whale. The gray whale we were able to sample as well. Because of Dr. Ravery's, which I'll just show there, he's in his office there. I've spent a lot of time this year with Stephen, and because he is so busy, a lot of these the results are pending on these necropsies. So we're hoping in the next 6 months to get results. A lot of the samples that we have, we're looking at biotoxins as issues. There's also vessel strike issues, ear entanglement issues. So piecing all those together, something that we're looking into. Just we've had them. Primarily these large whale deaths have been Central Coast and North Coast, which is quite interesting, and kind of leads into. We're not sure if there's a relationship, but in Alaska, as you all know, there been a huge large whale die-off, more than normal, up to probably 40 large whales this summer, spring and fall. So it's obviously a concern, and we were lucky enough to sample a lot of our animals. So that's kind of filtering in and feeding into that whole process as well with NOAA. And luckily, they're paying for a lot of our samples to get analyzed, which is very helpful as our budget is very limited. So I want to acknowledge just the whale watch industry, value that they play for the network. They're the eyes and ears out there. In particular, we had a couple of killer whale entanglements this year, which is quite rare. And the eyes and ears out there, we had A77, who about a month ago was seen with, it's on the right-hand side, or I guess, yeah, the right-hand side. And we were very concerned because it's quite rare to see killer whales entangled. So there was a big effort to try to relocate Transient that was entangled. And of course, we couldn't relocate the animal until about a week ago. And seen the animal 2 different occasions, and the animal is gear-free, so we're very appreciative of that. On the left-hand side, you're probably all aware of the Central Coast Cetacea Lab, which is a valuable member of our team. Worked with Hartley Bay First Nations to deal with this stranded young killer whale up in near Bella Bella Squally Channel, and did a fantabulous job of working with this animal over a tidal cycle, and then ensuring that the animal got free at the end of it. So another great success. We also had a killer whale, Transient again, off French Creek that was, and a lot of what we're seeing is, especially with Transient A77, they seem to have a bad affliction of actually playing with floats and ropes. So we're going to be watching this animal, and something that been noticed a number of times, this animal playing with this type of thing. So, and also the one Transient off French Creek was partially entangled, and Coast Guard were able to get out, and that was early summer, and help this animal and they're free. I just want to, we've had a huge number of large whale entanglements this year. A little more than normal, but the key has been that we've been able to get out there in quick manner. Within 24 hours is huge. Once these animals are seen trailing gear, that we can respond, and we have a very high success rate if we can get out there. We got, and I think this year Brian Gisborne has been huge with the west coast of Vancouver Island, and spotted probably most of the whales out there for us, and been a great help as well. In when we have been out to try to help these animals. Again, the Central Coast Cetacea Lab, Janie and Herman, and have been out with their trudging around with Hartley Bay First Nation with satellite tags and buoys to try to attach that to the trailing gear of these animals. It has been a challenge, but we have had a number of successes. And what I've got is a little bit of video of the Humpback River, or the Powell River Humpback Rescue, which is pretty, it's one of the first ones we've had in the inside, and it was a young animal, and it was actually anchored to the bottom. And I've got some cool video that I hope I can show. How do I do that? And it just shows the degree of, this animal actually had 50 prawn trap string anchoring to the bottom, and it was actually through the tail flute, tailstock, the mouth, and basically hogtied. Recreational fisher found it the night before when they were out fishing. And you can see this is probably only 24 hours, just the damage that polysteel rope has done. And this really helped getting this GoPro video of seeing how the whale was entangled. Because we thought, we didn't realize there was actually 3 wraps through the mouth to get hogtied. But we were able to then start from the mouth portion, release the tension on the mouth, and cut off the wraps around the tail. And this animal was exhausted. It's likely this animal would have, well, I don't know how much longer it would have been able to hold that strain up, but it was basically stationary. So we could approach it pretty close. Great help from the Canadian Coast Guard and fisher officers in Powell River. Again, it's just, it was just over a 24-hour period that we believe was there. And it's the first animal that we've had in prawn gear 3 years. It tended to go into neutrally buoyant rope between traps, so it's been a been a positive in terms of entanglements with here. And just this is again, that again. This is just some of the cutting that we did, and just shows the animals go off in the sunset. I also wanted to mention the value of our sea lion disentanglement team. I thought Wendy was going to be speaking, but just Marty and Wendy, the Vancouver Aquarium. I'm glad we don't have volume here because there was some swear words. But you know, again, the MERS is a huge Marine Education and Research Society. Jackie Hildebrand, Jared Towers, and Christy have been awesome in terms of coverage of entangled whales in key areas. And Christy was up in Fitzhugh Sound during a particularly bad salmon net fishery to make sure that if any animals were tangled, we were on. I'm probably missing lots of people, but we have, and again, Stephen Ravery and Hein really have been tremendous this year. I think I've spent more time with Stephen certain months than my wife, traveling around trying to, and you can call Stephen and he's ready in 12 hours. So it's just, you know, just the people in this room and within the experts that we have here really make the program work, and we're hoping that we can continue with these successes. And again, the 1-800 number is right here, and I'm done.
So if there's any questions for Paul while Stephen comes down. All back to the mic.
Yeah, all of that damage in the tailstock and even within the fluke, that polysteel rope, as anybody that's worked with, is fairly abrasive, and it's amazing how quickly it can work into the skin and blubber. And it's imagine a week later that animal would be in rough shape.
Our next speaker is Stephen Ravery, who's a wonderful asset to not only our students here at UBC, but also to DFO, Vancouver Aquarium, and to many others, including many of our colleagues in the U.S.
Thank you, Andrew, and thank you, Pamela, for the opportunity to present at this session. The focus of this talk is on our work on reviewing post-mortem findings of stranded killer whales from 2004 through to 2013. And obviously this would entail quite a multidisciplinary and multi-institutional effort. And I just wanted to acknowledge the people that we've been working with quite closely to try and compile and assimilate this data and then try and put it in context of what might actually be going on in some of these populations. We really value the opportunity to go out and conduct these necropsies. It really affords us a unique opportunity to understand some of the natural history of the animals. By doing postmortem examinations, we can actually assess a specific cause of death. This is through our pathology training, but also look for diseases or disorders that may not necessarily contribute to death, but can impair health. And by that, I mean there are certain conditions that may impact the ability of these animals to successfully reproduce or to successfully forage, and many other factors. And these are increasingly our areas of particular interest to us as a community trying to investigate and understand some of the disorders in these animals. As well, you'll see in the study that what we've done is broken down individual findings by age class, and we find that this really helps us refine specific disorders or diseases in different age classed animals. So perinates or neonatal animals, calves, adults, and so on. Just some of the efforts that are currently underway in terms of nutritional condition, looking at infectious diseases. We're now trying to adapt some of the most advanced molecular sequencing and really collaborating with some of the preeminent laboratories, not only in the United States, but throughout the world. Over this 10-year span, we had 53 strandings, and just to put this in context, we've done a study previously looking at all stranded killer whales. Initially, our initial focus was worldwide to try and garner some understanding of population dynamics in different regions. We were able to recover or identify 242 strandings since 1940 through to 2000, about 10. And so that number has since increased. By dedicated funding through NOAA and Department of Fisheries and Oceans, we've actually increased our efforts to try and attend to these animals that do strand and have been able to conduct necropsies on 53 individual animals. And you'll see that only 27 of them we've identified as having a specific disorder or disease. In many instances, some of these animals can present with fairly advanced autolysis. Some of them are just inaccessible. There are many things that may hinder our ability to actually conduct a very thorough and comprehensive necropsy. I should emphasize that this doesn't just focus in British Columbia, but actually we have animals that have been identified from Alaska right through to California and even in Hawaii. With the breakdown, a number of strandings according to year, and this is again over a 10-year span. You can see that the highest number was in 2011, and this was due to 3 individuals in particular up in Alaska that had swum into a fairly deep river and subsequently died of, we believe, emaciation. It's sort of an extraliminal type of process. By categorizing the diseases, this further helps us to understand some of the mechanisms that might be involved. And these are just general categories. We would use these if we're looking at Vancouver Island marmots or any sort of animal. We just break them down into general disorders. So there's nutritional metabolic. This would include malnutrition, deficiencies, or toxicities, as well as metabolic disorders. We've identified even in entangled humpbacks where these animals are straining to the extent that their muscles are starting to deteriorate. They get a lactic acidosis and a variety of sequelae related to that. Trauma has been identified as human interaction as well as non-human interaction. Extraliminal, we would envision these as animals that are outside of their normal range. So again, going back to these 3 killer whales that were up in Alaska, they were in a freshwater river for 6 weeks, and then subsequently died, we believe, because of starvation. Congenital, degenerative, toxic, and undetermined. And unfortunately, the undetermined category is one that is our #1 conclusion, and this is largely derived by reviewing, each case is reviewed by 5 different pathologists, and you can imagine the expertise that's involved in reviewing this and the input that's sought. And ultimately, this is sort of the culmination of that effort. There were 6 animals in which we were able to find a conclusive diagnosis, and I should emphasize that we're even further trying to define these as the proximate and immediate causes of death. So proximate may be what may have set the animal up to a particular disease, whereas the immediate may be something that's supervening or secondary to that inciting cause. So in this calf mortality, you see that infectious disease. This one from Alaska was actually because it had ingested a fish hook and, oh, I'm sorry, I might have skipped a slide. Oh no, this is one where an animal had ingested a fish hook that impaled the pharynx or the back of the throat, subsequently resulted in a septicemia or bacteremia. The second animal on the list from California is quite interesting. This is a little neonate, offshore neonate, that was found stranded in Santa Barbara, and it turned out that it had Salmonella Newport septicemia. This is something we typically associate with terrestrial animals, poultry and cattle and so on. And the big question is, if an animal is that far offshore, how is it actually being exposed to this bacteria and then subsequently resulting in the death of this particular animal? What's a little bit more disconcerting is that it's very likely that infection was in utero, so the mom was probably exposed, the bacteria localized to the placenta and then was transmitted to the developing fetus. Mentioned the metabolic, and this is one that we're particularly interested in. These are animals, and this one in 2010 from BC was actually an animal that was recovered by Souk. And this little calf, we are able to take post-mortem heart blood and submit that to a lab, and they can quantify the amount of immunoglobulin that's present. So that first suckle that a calf or a neonate takes is where the mom is transferring immunoglobulins to the little calf, and those are assimilated into the calf. And we actually have techniques of measuring that. And what we're finding increasingly is that that isn't occurring in some of these young calves. And so they're not getting that protective barrier, and they're also losing out on the nutrients in that first colostrum bolus. Moving on to the next age class, again, trauma is something that we weren't really appreciative of when we first started to look at this, but increasingly something that's becoming of concern, and trying to look at it from a forensic perspective, whether it's a primary cause of death or whether these animals may have other underlying diseases that are predisposing them to some sort of physical injury. And then the adult, this animal from 2004 with the mesosalpinx, these are essentially benign smooth muscle tumors within the reproductive tract. And what was interesting is that this animal was also culture positive for Brucella. And Brucella is a bacteria that historically has been found in dairy cattle and a variety of other species and can cause significant reproductive loss. The brucella in these animals in killer whales is different to the one that we find in terrestrial species. But in fact, there have been case reports of the marine mammal brucella infecting people, resulting in quite significant. And then you can see the spectrum of other disorders that have been identified, the metabolic, beach casting. And again, this would go back to an animal where if it's found on shore, you can imagine the weight of the animal is actually compressing the muscle. And as a consequence, the blood flow can't perfuse through those areas. You get a loss of oxygen, a buildup of lactic acid, and essentially the muscles become solubilized. So these are something that's of tremendous importance to us. And then this is just to round out the last of the group of animals. And again, you can see the undetermined is something that's quite significant in these particular cohort. What we need to do, or what would we need to do to enhance investigations in the future? And this is really drawing on a lot of the work by Mike Moore and Bill McClellan in the Northeast, with the Northern Right Whale effort. And their reports are actually immaculate. They're about 30 pages long, and they really try and tie in and integrate what's going on in the individual animal, the population, and the environment. And that's where I'm hoping that we can start to direct our efforts with a lot of the work that's being done with the killer whales. Faster response, and we're getting really good in terms of mobilizing and identifying people. It's really a team effort out there to try and attend these animals and do a necropsy. We're really interested in looking at some of these perinatal animals. It looks like the perinates or the neonates, very young animals, and then the very geriatric ones are the ones that are dying out in the population. And it would be nice to get a better handle on what's going on. We're still having some difficulty in terms of refining ages of these animals. So being able to discriminate between subadult and adult would also be of tremendous value. And again, quantifying evaluation of nutritional condition. And this may tie into the work that Lance and John Durbin are doing with the drones and trying to better quantify the nutritional status of the animals, and then also looking at the reproductive status as well. So our next steps are really of particular interest, looking at the clinical and post-mortem findings in the context of contaminant loads. I mentioned earlier about this study that was published with Michelle Barbieri, University of California at Davis, the US Navy, NOAA, DFO, and ourselves, where we were able to look at 242 strandings, and you can see they're mapped out on the map on the right. We'd like to actually go back and look specifically at the distribution and findings of the southern resident killer whales to better understand what might be going on in the current population, and then again looking at these environmental factors much more closely. So again, a lot of this effort wouldn't be available, or we wouldn't be afforded the opportunities to go out without the assistance of DFO and NOAA. Then there's staff here at the university, Animal Health Center, CDOC Society, as well as many of you who are out in the field and reporting these to the Marine Mammal Response Program, and then a list of collaborators. Thank you very much.
Do we have any questions? If not, you can find Stephen down here in the front. We're going to take a 15-minute break. Please head back to your seats at 11.